Polyethylene is the most common kind of plastic on Earth, and its large accumulation is an issue of great importance. Please join us to address the degradation of polyethylene, including current knowledge and future directions.
Here are all the sections we are going to discuss. First, we will have a brief introduction to polyethylene, highlighting the importance of its biodegradation. Next, we will summarize the polyethylene degradation by gut microbiomes and current bottlenecks, which is one research hotspot in this area. After that, we will propose another new pathway for polyethylene degradation based on literature review. Finally, we will speak to some future polyethylene research directions.
Polyethylene is the most common plastic on Earth. In 2018, it made up 32% of all plastic produced. Disposal of polyethylene is difficult and must be addressed to handle the large quantity of it globally. Primary disposal methods include recycling, which is relatively rare, and burning, which produces greenhouse gas emissions. Most polyethylene ends up in landfills and oceans, where it degrades slowly into micro and nanoplastics that pose human health hazards. An alternative, sustainable way to handle polyethylene is needed.
With the efforts from biologists in the past several decades, we have identified some ways to achieve biodegradation of polyethylene. In 1980, the first bacteria capable of degrading polyethylene was isolated, which was named Fusarium redolens. From then on, polyethylene degrading microbes were discovered one after another from various environments, including garden soil, rivers, and so on. Importantly, in 2014, researchers found that the gut microbiomes from worms have great potential for polyethylene degradation. Here, mealworms are eating plastics as their feedstock are seen. This is an amazing and promising way to sustainably degrade polyethylene with no disposal of toxic chemicals to the environment. This image is from a paper published in 2014, in which researchers discovered evidence showing the potential of polyethylene degradation by mealworm gut microbiomes. Several follow-up studies have successfully identified bacterial species in gut microbiomes responsible for polyethylene degradation. This table summarizes 3 recent works that have identified different bacteria species. Despite these isolations, the mechanism of polyethylene degradation by these bacteria species is unknown. In other words, the metabolic pathway is of special interest.
In a proposed chemical polyethylene degradation pathway, the first step is to add radical species on polyethylene polymers so that these high-energy groups can be degraded by subsequent enzymes. It is unknown how bacteria and gut microbiomes achieve this first step that will be referred to in this video as biodeterioration. Typical chemical processes involve high-energy reactants like ultraviolet rays or high temperatures, but it is believed that these methods cannot be used in gut microbiomes. The PE polymer is then degraded into its monomer by a series of proposed enzymes, including lacase, manganese peroxidase, and so on. This second step is called biofragmentation or depolymerization. Finally, the monomer will be assimilated by bacteria and enter the bacterial central metabolism, which finishes the assimilation and mineralization of polyethylene and completes degradation by bacteria. This pathway is a putatively proposed theory and has not been confirmed by experiments. The real PE degradation mechanism by gut microbiomes largely remains unknown.
To try and discover the actual polyethylene degradation pathway, it is important to consider first why it is so difficult to degrade. Polyethylene is very stable and nonpolar, and carbon-carbon bonds are difficult to break. To assess whether or not we are correctly building a pathway, let us look at a much better characterized plastic, polyethylene terephthalate. This plastic has esters which makes it susceptible to breakdown by esterases. Even though there are chemical differences between polyethylene and polyethylene terephthalate, both plastics started out as chemically stable and a mystery to degrade. It would reason that we can use similar approaches that were used for polyethylene terephthalate to polyethylene to discover and use degradation pathways. We can use the broad steps for polyethylene terephthalate degradation to try and break down polyethylene. The first step polyethylene terephthalate breakdown is to isolate repeating units. Because of the size of polymers that have many repeating units like plastics and bioplastics, it is likely that polymers are broken down into monomers extracellularly first, so transporters and ports importing material to the cell for processing may not be a concern until later. Unfortunately, after this crucial first step, the chemical similarities between polyethylene terephthalate and polyethylene seem to end, since ester bonds and phthalates are not chemically possible from the polyethylene starting material. Degradation of polyethylene is far from over after initial polymer breakdown. Many steps have been well characterized in the polyethylene terephthalate breakdown, and it is likely the same will be for polyethylene. In the same vein, many steps have yet to be confirmed in terms of polyethylene terephthalate breakdown, which can be due to different microbes taking slightly different approaches, or needing to work in consortia for adequate breakdown. The same may be said for polyethylene when the pathway enzymes begin to be elucidated fully. It is important for researchers to be aware that there are many different strategies that may ultimately be taken by bacteria for complete breakdown to occur.
Before we can assess the individual steps after initial breakdown, we must step back and determine how to degrade polyethylene, the polymer, into ethylene, the monomer. To do this, bioprospecting may be the answer. Bioprospecting involves taking microbes from a wide variety of sources and assessing their phenotypes and qualities. For plastic degradation, it is best to search in areas that may have some plastic contamination, since it will be beneficial for bacteria to have degradation machinery to best survive in the contaminated regions. Some areas proposed in literature include cow dung and stomachs of bugs like mealworms. One other source poses an interesting possible place to find bacteria: hydrocarbon sources. Hydrocarbon degradation has been traditionally applied to and studied in the cases of oil spills, where bacteria like Marinobacter hydrocarbonoclasticus have been unsung heroes in cleanup practices. The bacteria thrive on the nonpolar molecules that make up oils. Butane, a fuel degraded by hydrocarbon consumers, is chemically similar to polyethylene. The compounds that hydrocarbon degraders and polyethylene degraders consume contain the same single carbon-carbon bonds. Therefore, applying hydrocarbon degraders to plastics like polyethylene may be a potential source of degradation.
We will complete this video with a few future research directions responsible for polyethylene degradation in worm gut microbiomes. One could expect a functional genomics study should help elucidate the genes and their coded enzymes that are responsible for polyethylene degradation. Based on the latest CRISPR techniques, the functional genomics by gRNA libraries could be easily constructed to achieve a full genome screening. An example is shown in Nature Microbiology, where authors evaluated each gene in Vibrio naturigens by high-throughput CRISPR screening coupled to NGS. Similar approaches could be adopted to screen candidate genes that might participate in polyethylene degradation in those isolated polyethylene-degrading bacteria.
Another interesting facet of polyethylene degradation by worms is that a recent study shows the importance of worm saliva in degrading PE. Two enzymes belonging to the phenol oxidase family were identified. This suggests researchers should not just focus on the gut microbiomes but also pay attention to the worm itself.
Bioremediation is an important consideration in the goal to not only clean up plastic pollution, but also to upcycle them into something beneficial. This is a rapidly growing field and is on the agenda for many Department of Energy national laboratories. As for the upcycling of polyethylene, metabolic engineering might play an important role. Although the pathway of polyethylene degradation has not been elucidated, we could refer to the successful precedents of polyethylene terephthalate upcycling. Shown here is a recent study focusing on converting polyethylene into vanillin, which is one target chemical in metabolic engineering. Knowing that polyethylene will eventually enter the central metabolism of some bacteria, one could expect that adopting similar methods will also promote the upcycling of polyethylene to other value-added compounds.